Hello, everyone. My name's Asad. I'm a respiratory physician, or as you might call in the US, a pulmonologist in Manchester, England. I have long COVID, which links in nicely with today's discussion. So first up, we've got Dr. Mark Walsh Md, who will take us through his management of acutely ill COVID-19 patients. Dr. Walsh is an emergency physician and internist who has studied trauma-induced coagulopathy, sepsis-induced coagulopathy, and more recently, COVID-19-induced coagulopathy over a career spanning more than 40 years. He is a staff emergency and internal medicine physician at St. Joseph's Regional Medical Center in Mishawaka, Indiana, and also a clinical assistant professor of medicine at the Indiana University School of Medicine, Notre Dame. So without much further ado, over to you, Mark. Welcome.
Thank you so much. It's an honor to be here. The title of my talk is COVID-associated coagulopathy, the clot thickens or not, threading the needle, complying with evidence-based medicine while riding the roller coaster on the cytokine storm. I'll be sharing this with Dr. Pretorius and Dr. Kell. In about 20 minutes, when I'm done, they will take over. My objectives are to describe why patients with COVID-associated coagulopathy clot and bleed at the same time, how to anticoagulate in and out of the hospital, review examples of thromboelastographic and plasma-based coagulation tests' ability to provide goal-directed anticoagulation therapy for COVID-19 patients, and introduce our United States collaboration with Drs. Pretorius and Kell and their foundational work, which helps us explain the pathophysiologic foundations for the short-term and long-term complications of COVID-associated coagulopathy. So before we start, I want to talk about the TEG, or thromboelastography. Basically, it's a cup into which you put 0.3 cc's approximately of whole blood. And this cup turns at about 4.5 degrees every 10 seconds. And as the blood clot forms and attaches to a pin that's dropped in, that's attached to a transducer, on the y-axis, you have millimeters of displacement of this pin as the pin and the cup eventually move at the same rate as the blood clot forms. And this curve looks like a shovel, where the shovel is reflected by the R value, the alpha angle reflects fibrinogen. The maximum amplitude of this shovel reflects mostly platelet function with interaction of fibrinogen, and the lysis of that clot is reflected by the decline in the thickness of this clot, or this curve. And basically, this looks like a shovel. And that all COVID patients have a thick TEG with a short handle, large alpha angle, fat MA, and no lysis. Imagine blood of a COVID patient like dirt, hard dirt, and this shovel won't move it. You put these patients on heparin, and as the cytokine storm declines, the handle gets long, that is, the R gets long, which reflects the PTT. The alpha angle will get flat, and in severe cases of hypocoagulopathy of COVID, they get thin maximum amplitudes and high lysis. The thick clot, the prime example of hypercoagulopathy is what we call fibrinolytic shutdown, where there's no lysis in these patients and they have a thick blade on their TEG shovel. So we have a lot of experience. Since 2007, we've been doing TEGs with platelet mapping. We've published widely on this. So when the pandemic came, we were primed because in Europe, there had been exhortation from Donat Spahn and many others from the Europeans to use TEGs and the European counterpart where the pin rotates, the ROTEM, to guide goal-directed coagulation with TEG and ROTEM, and not so much with the common coagulation tests, fibrinogen, prothrombin time, and partial thromboplastin time. And so basically this complicated picture of the endothelium, which you have the thrombomodulin thrombin switch complex, which judges in the direction in which these patients will go on this teeter-totter of trauma-induced coagulopathy, of anticoagulation pathway or hypercoagulation pathway. Once platelets are activated in patients that have hyperfibrinolysis, activated protein C mediates fibrinolysis. But at the same time, there's a counteracting antifibrinolytic movement that causes diffuse fibrin deposition, aberrant procoagulants that causes clotting and bleeding. And this, the peripatetic Jeffrey Dobson from Queensland, Australia, has described this as the clot thickens or not as a mechanism for trauma-induced coagulopathy. Because the endothelial cell lining covers a surface area of up to 7,000 square meters, the size of a soccer pitch, an NFL or Australian rules football field. And this thrombomodulin thrombin complex and fibrinolytic system can switch from fibrinolytic to antifibrinolytic phenotype within seconds to minutes. Finding the right balance and restoring hemostasis in vivo is an extraordinary challenge, not just for trauma-induced coagulopathy, but also for COVID-associated coagulopathy. Par Johansen Jacob Stensballe and Sissi Ostrowski at the Rikshospital in Copenhagen, Denmark, have described the endothelial dysfunction, looking at the endothelium as if it were an organ unto itself, as shock-induced endotheliopathy or SHINE. And if you look at this within the context of trauma and COVID, you can see that the endotheliopathy of TEG-mediated identification along the spectrum of trauma-induced coagulopathy or COVID-associated coagulopathy can range from the physiologic on the left hand to the shutdown in the middle to the hypocoagulopathic to the uncommon but lethal hyperfibrinolytic phenotype based on the TEG, which reflects the underlying endotheliopathy. So here's a perfect example of how it works. This patient was admitted last July on room air with mild COVID. He quickly deteriorated. We anticoagulated him, transferred him to the intensive care unit 5 days later, and he was on BiPAP. He was moderately ill, not severely ill, and we gave him therapeutic heparin. Look at the width. The maximum amplitude is 70.7. Look at the short handle. This is the thick shovel TEG. He improved, and the heparinase sample, that is the sample that shows with no heparin, just 7 days later on the 30th, you see a long R, a normal R, and a maximum amplitude that's 55. It's normal. This TEG reflects what's going on in the endothelium, how the anti-inflammatory and anticoagulant effects of heparin help this patient early in the course of his disease. So for clinicians trained in using evidence-based medicine approach, we find ourselves forced to practice without data. Some guidance documents from around the world early on in April of 2020 recommended full therapeutic dose anticoagulation in these patients. These are colleagues, Jared Levy, I've published with, Gene Connors is well respected. We were struggling on how to approach these patients who clotted, because the COVID-associated coagulopathy teeter-totter is just like trauma. Based on TPA levels, plasmin, Pi1 and TAFI levels, based on fibrinogen and D-dimer levels, this teeter-totter shifts between the thick tag tracing on the right and the thin one on the left, which reflects the underlying endotheliitis at the level of the endothelium. Our friend Jaco Thachil proposed in May 2020 in JTH that fibrinogen may have a protective mechanism in protecting the host from the effects of the spike protein in the virus. Yet it's a double-edged sword. And he first described this teeter-totter of acute phase response to thrombosis, where the fibrinogen goes up initially as thrombosis occurs, but then quickly will drop into the thrombohemorrhagic state. Dr. Pretorius and her colleagues have very cleverly described the cytokine roller coaster of early on the left, the acute stage, where the thick tag is reflected by the thrombotic risk. And then during the cytokine storm, which varies like a hurricane, you have the subacute stage that can either be physiologic or even hypocoagulopathic. And then the remote period of recovery, which in patients with long COVID may last months. So the goal of treating what I have called and what our colleagues call upside down trauma is to prevent bleeding and clotting during an era bereft of evidence-based medicine. We published a few papers from April 2021, and I'm going to focus the remainder of this talk on our paper where we proposed a protocol, not a full anticoagulation, but TEG-guided adjunctive anticoagulation for patients only with unfractionated heparin. In preparation for this, we published an editorial with my mentor, Professor Kwan, a lifetime member of the ISTH, and Professor Matthew Neal, a critical care trauma surgeon, to describe the endotheliitis-induced immunothrombosis caused by the SARS-CoV-2 infection that represents a crosstalk not just between immunity and coagulation, but between medical and surgical specialists. And it leads to a spectrum of inquiry where randomized controlled trials are informed by some small hypothesis-generated observational study using precision-based medicine, specifically the TEG, to describe the clinical fibrinolytic phenotype, which is that of shutdown. We defined COVID-associated coagulopathy as upside-down trauma in this paper, where this upside-down trauma, as we state, quoting from this paper, can be characterized whereby the upside-down trauma is where COVID patients present in a hypercoagulopathic state, but may quickly evolve into a hypercoagulous state, as opposed to the typical progression of trauma, where they come in hypocoagulable and then very quickly, with resuscitation, blood products, source control of the bleeding, become hypercoagulable and a risk for DVTs and pulmonary embolisms. The hypercoagulability of COVID-associated coagulopathy can quickly be modulated by the vagaries of the cytokine storm. And as this storm recedes, these patients become exquisitely sensitive to anticoagulation, causing them to bleed. Hence, these patients are said to bleed and clot at the same time. This is our paper. These are the authors, and you'll see Dr. Kwan, my mentor at Northwestern, Dr. Neal, and the trauma surgeon, the legendary trauma surgeon, Gene Moore, who has worked so long at Denver General. He now has the Ernest E. Moore Shock Trauma Center named after him. This paper was published in mid-July 2021. In 2019, after a raucous discussion about tranexamic acid in trauma that Beverly Hunt kindly modulated and calmed everyone down, Dr. Neal was discussing TEG, sepsis, and trauma while Dr. Moore ordered him another beer. And in the end, someone needed to get this guy a beer. Our algorithm is the following. Does this ICU patient have a thrombosis? If so, to the left, they get the intermediate dose without a bolus. If they do not have a thrombosis, if they do have a thrombosis, they get to the right, no bolus and 14 units per hour kilogram. Further adjustment is determined by the APTT goals, which are modest compared to normal algorithms, and TEG algorithms, which we've adopted from patients on ECMO. So basically, intermediate dosing for non-clotting patients, therapeutic dosing for clotting patients, no bolus. APTT goals are modest. We've adopted this from ECMO. And the ECMO goals are also followed with fibrinogen, which we like to keep above 200, and R values that reflect the APTTs often for our patients on heparin. The take-home points of our study were in the pre-protocol group when we adhered to the guidelines recommending full anticoagulation for our patients. We had what I consider a catastrophic number of patients who bled. This was reflected by Dr. Kotze in his editorial, which we will describe subsequently, where up to 30% of some centers were reporting this level of bleeding. Clearly, we had to do something. So we came up with our protocol, TEG adjunctive protocol, and in the post-protocol non-bolus group who were treated with heparin, they bled at a rate of 2%. Post-protocol non-bolus patients required less blood products. The TEGs and fibrinogen D-dimer common coagulation tests, APTT, predicted bleeding and thrombosis when studied with linear regression analysis. And the same thing for prediction of bleeding when patients who had long Rs, prolonged APTTs, low D-dimer, and low fibrinogen. And the opposite for those who had short Rs, thick MAs, elevated D-dimers, and alpha angles for prediction of clotting. Here's the first group of patients treated. The 31.4% was an absolutely unacceptable rate of bleeding with an acceptable 2.3 afterwards. Notice the use of packed cells was reduced significantly, as well as fresh frozen plasma in the second group. Looking at the bleeding and non-bleeding patients, again, a reflection, a confirmation of our study between the protocol and non-protocol groups regarding packed cells, platelets, and fresh frozen plasma. Using multivariate linear logistic regression models, we showed that these intercept had a very high correlation between bleeding with a long R, low fibrinogen, low D-dimer, and elevated APTT. About the same time, about 3 weeks after our paper was published, Hugo Tenkate published Surviving COVID-19 with Heparin. He reiterated the high incidence of bleeding with heparin and exerted caution with its indiscriminate use. However, he noted that the multi-platform 3 trials, the Active 4, the REMAP CAP trial, and the ATTACK trial, all showed benefit for patients with moderate illness. In the executive writing committee was Dr. Neal. Even up to date, a few weeks later, after our paper, consented that based on the judgment of clinicians, that intermediate or full dose was indicated for patients with moderately ill COVID-19, based on these 3 trials. Basically, 27% of people treated with intermediate or full dose anticoagulation needed intubation with a reduction in mortality of 4%. These 3 trials did not have the TEG, which enabled us to give anticoagulation to these moderately and severely ill patients without hemorrhagic complications because they did not have the intense monitoring and the personalization of dosing that we have. Therefore, we conclude that our single center observational demonstration of the use of therapeutic unfractionated heparin guided by adjunctive TEG may justify the use of precision-based medicine approach to providing safer anticoagulation to severely ill COVID-19 patients. Just about the same time, it's September 1st, as my New England Journal hard copy arrived, Dr. Moore, myself, and others published a letter to the editor exhorting emergency physicians to become familiar with the TEGs. And the peak of my career is when someone sent me this text of Dr. Neal showing our algorithm at the ISTH devoted to COVID-associated coagulopathy. And the star of the show was none other than Dr. Pretorius there at the end, with Gerald Levy on the left and Paul Grobbelaar in the middle. I've shared co-authorship with all of these patients. So how does this happen? And in conclusion, it happens because in this period of a dearth of evidence-based medicine, you need treehouse medicine, where emergency physicians, critical care doctors, interventional radiologists, hospitalists, infectious disease specialists, hematologists, clinical pharmacists, who are crucial, nursing and lab, we're all like a treehouse. And the antidote to evidence-based medicine lack is point-of-care bedside medicine, or what we call treehouse medicine, based on the foundations of excellent researchers that give us the eternal vigilance given to us by Dr. Kell and Dr. Pretorius. I'd like to thank these people who helped make this possible. Now I've done this in 20 minutes. I hand this over to you, Dr. Asad. Thank you.
Well, Mark, thank you very much for a most engaging talk and some really, really impressive results there in your paper. The key message I gathered from your talk is that we need to be practicing personalized precision medicine using viscoelastic methods at the bedside if we are to treat COVID-19 associated coagulopathy correctly. My question is, given that TEG and ROTEM have been around for decades, why are you one of very few clinicians employing them in your practice? I've heard hematologists who are meant to be experts in clotting dismiss the utility of these measurements. It's baffling.
Well, we only have an hour today, but simply put, science moves slowly. And I recall that the same hematologists were not in favor of using this for liver transplantation. The same hematologists were not in favor for using this for heart surgery. The same hematologists didn't want to use this for trauma, resuscitation. The same hematologists didn't want to use this for obstetrics and gynecology. The same hematologists didn't want to use this for COVID-associated coagulopathy. But for the first four that I've mentioned, we now have evidence-based medicine guidelines that suggest that each of the four areas using viscoelastic testing is possible. The second thing is these tests have become more robotic, if you will. We have cartridges for both the TEG and the ROTEM. We have the Clot Pro and the Quantra. We have now multi-channel type assays that lend themselves to batch analysis. I think the future is bright, and I would ask our hematology colleagues to look at the past and remember that the past is prologue, and that within the next few years, hematologists may be using these more often than they had thought, and that the COVID pandemic has allowed us to explore this with greater intensity than we ever imagined.
Thanks. Thank you very much, Mark. We live in hope. I've got one more question for you, if that's okay. So knowing the key role of the platelet in COVID-associated coagulopathy, did you ever consider adding in antiplatelet therapy?
Yes, we did. I think Dr. Pretorius's group was very aggressive in this. There are groups who have. We did not because we wanted to do one thing at a time. And we were not able to monitor platelet function because we could not get the turnaround time with TEG with platelet mapping. So for scientific consistency, we just used the TEG 5000. We did not have the TEG 6S. And I think that there are a few other groups that also did, that added clopidogrel and aspirin with heparin, but we didn't want to expose the patients to bleeding. And then again, in deference to evidence-based medicine, I didn't want to cause more complications. So we just basically did one thing at a time, excuse me, trying to adhere to Occam's razor, which is pluralitas non est ponenda sine necessitate. Plurality is not to be posited without necessity. I did one thing, and I'm going to let Dr. Pretorius and Dr. Kell take care of the platelet.
Okay, right. Well, I guess that's the perfect note to move on to the next part of the talk. Our next two speakers have somewhat, should I say, iconic status in the long COVID and ME communities because of their path-breaking work identifying clotting abnormalities in these conditions. I have to say that I owe a lot of the improvement in my condition to the work that they have performed alongside their pioneering clinical colleague, Dr. Jaco Laubscher. I've got to know them really well over the last year, and really all that motivates them is helping the millions who are suffering in silence. They really are a rare breed of scientists. The first up, we've got Professor Resia Pretorius, who is the head of department and distinguished research professor in the Physiological Sciences Department within the Faculty of Science at Stellenbosch University in South Africa. Her research focus is coagulation and inflammatory markers and their role in inflammatory conditions like type 2 diabetes, rheumatoid arthritis, and Alzheimer's disease, and more recently, both acute and long COVID, as well as ME. She will be discussing the latest preprint from her group. Over to you, Resia.
Hello, everyone. Thank you, Asad. And what a pleasure to sharing this platform with both Mark, yourself, and also with Doug, my longstanding collaborator. Our focus has been identifying inflammatory molecules in circulation that might be involved in and drive pathological clotting. And we have also focused our research endeavors over the years on studying the effects of increased circulating inflammatory molecules and how they interact with cells of the hematological system. We focus in particular on platelets and red blood cells, as well as on the clotting protein fibrinogen. We are also interested in identifying novel inflammatory molecules that might play a role in persistent symptoms of long COVID. Over the years, we have published numerous papers that show the importance of inflammatory molecules in circulation and their role in abnormal clotting. We are particularly interested in platelet signaling and the role in abnormal clotting. Platelets in circulation play a critical role in healthy blood clotting. However, they can become overstimulated and can drive pathological blood clotting if there are inflammatory molecules in circulation. This can also happen in the presence of viral infections, where platelets can act as important signaling entities. There is also a complex relationship between receptors on platelets and endothelial cells, where circulating biomarkers may bind to. As we now know, damaged endothelial cells and platelet hyperactivation are central in many pathologies. Our research group have shown these pathologies also in acute COVID in published papers. In the context of COVID-19, platelets are therefore central in immune activation and general coagulation pathology, and they can form various complexes with other circulating blood cells, as shown in this illustration. Platelets can form complexes with T cells, monocytes, neutrophils, macrophages, and obviously also with themselves. This is known as platelet clumping. We are also interested in pathological blood clotting involving the main clotting protein, fibrinogen, which is a soluble protein in circulation. If you focus your attention to the two cartoons shown in figure A, on the left is a healthy protein structure, which has many alpha coils and few beta sheets. However, in the presence of inflammation, oxidative stress, and circulating inflammatory molecules, this structure changes, where the alpha coils untwist into beta sheets, as seen in the cartoon on the right. In plasma clots from healthy individuals, these clots have more alpha coils and have the ultrastructure as shown in figure B. Various inflammatory conditions, including cardiovascular disease and diabetes, have a clot structure similar, as shown in figure C. In early 2020, we looked at blood clotting in acute COVID using the scanning electron microscope. We found that platelets are damaged and clumped together and attached to red blood cells. The clotting proteins that are supposed to be soluble form spontaneous microclots. In the micrographs on the far left, you can see platelets that are hyperactivated. Note the yellow arrows. In the middle are micrographs where platelets are attached to red blood cells. Note the white arrows. On the far right, you will see micrographs of spontaneously formed microclots that even glue together red blood cells. Note the blue arrows. Now, with this ultrastructural pathology in mind, we looked at platelet-poor plasma and exposed it to our fluorescent marker, thioflavin T, which binds to open hydrophobic areas in proteins. This marker was first used to identify amyloid protein in the brains of Alzheimer's disease patients. However, in 2014, our research group, together with Kell, discovered that thioflavin T also binds to misfolded fibrinogen and fibrin proteins during pathological blood clotting. Here you can see the differences in platelet-poor plasma protein structure in acute COVID-19 patients, where we compared plasma smears of acute COVID patients to that of healthy and diabetic patient plasma samples. We exposed our plasma to thioflavin T, as mentioned previously. In healthy plasma, the various clotting proteins and also fibrinogen are soluble, but during inflammation and clotting disorders, these molecules might become damaged, resulting in insoluble microclots in circulation. Here you can see the microclot formation demarcated as the green signal from thioflavin T. In this COVID experiment, we found small areas of nearly undetectable abnormal clotlets that are present in plasma of the healthy individuals, as can be seen on the micrographs on the left. More abnormal microclots were noted in plasma of individuals with diabetes. This is in line with our knowledge of the presence of hypercoagulation in type 2 diabetes mellitus, which is indeed well known. What was, however, quite significant to us was the extent of the abnormal microclots in acute COVID-19. See the micrographs on patients of COVID-19 on the right. Also note the scale bar of 10 micrometers at the bottom of the micrographs. However, as microscopy results are quite difficult to quantify, we suggested a microclot and platelet grading system based on various stages of severity of the microclot activation we noted, and also based on the stages of various platelet activation. Here are examples of our platelet grading system. In our experiments, hematocrit samples were exposed to fluorescent platelet markers. CD62PE, which is conjugated and a pink signal, that is the marker of P-selectin. And it can either bind to P-selectin now, can either bind to the membrane of platelets, or it can be found inside platelets, or it can be a free molecule. But the platelets that are shed and shedding their content you could see the signal of this marker. We also used PAC1, which is the green signal that identifies platelets through marking of the glycoprotein 2 beta 3 alpha on the platelet membrane. Stage 1 are examples of platelets from healthy or control samples with minimal activated platelets. In healthy blood, platelets are small, round cellular entities with few pseudopodia visible. And this is only visible due to contact activation after the blood is drawn. The plate shows platelets spreading and the beginning of clumping as seen up to stage 4. In the right plate, we show microclot and platelet clumping together from stage 1 with minimal formation until the more severe clot formation of the platelets and the membranes clumping together where platelets are hyperactivated. Here is our microclot grading system just again, where we show stage 1 with minimal microclot formation in healthy controls. Stage 2 and even stage 3 might be seen in various inflammatory conditions like type 2 diabetes. And for comparison, I again included the diabetes microclot micrograph shown a few slides back to just compare the actual samples from a diabetes sample. Plasma will progress to the presence of severe microclotting in stage 4. We also suggested that the various stages can be used in a numerical scoring system that might be used both for acute COVID-19 and long COVID. So with our research understanding of the older beta and delta variants, which this research was based on, and as that gained momentum, arrives the new kid on the block, Omicron. It soon became clear that at least in South Africa, Omicron was not as virulent as the older variants. Our team of international collaborators, under the lead of Mark, wrote a review paper earlier this year with title Immunothrombotic Complications of COVID-19, Complications of Timing of Surgery and Anticoagulation. You might note some of the names of the renowned researchers where that also Mark noted previously on this publication, including Ernest and Hunter Moore, Becky Neal, and then of course Mark himself as corresponding author. In this review, we discussed the hemostatic derangements that occur during acute COVID-19 infection and discuss and also argue that hemostatic derangements affect not only the timing of surgery for emergency procedures, but also the incidence of post-operative hemostatic complications related to the COVID coagulopathy that we have been noting. Traditional methods of prophylaxis and treatment of arterial and venous thrombosis after surgery require a tailored approach, guided by understanding of the pathophysiological derangements of the hypercoagulable COVID-19 patient. Likewise, a prolonged period of risk for developing hemostatic complications following hospitalization due to COVID-19 has resulted in guidelines for differing societies that recommend varying periods of delay following this COVID-19 infection. In our review, we also described the pathophysiological foundations of the COVID coagulopathy again, and the need to establish a continually evolving guideline regime for timing of elective procedures. As part of the discussion and a plea for focused guidelines, we showed examples of microclots from 3 groups and the corresponding TEG traces. And those 3 groups are from the Delta variant, Omicron, and then obviously from a healthy individual. We were the first group to report on reasons why patients with Omicron do not typically suffer from significant complications, or at least not as was found in the more virulent variants. Also interesting, at least in South Africa, significantly fewer patients landed up in hospital than with the older variants. Also, the symptoms were changed quite radically. Fewer significant complaints were noted, including shortness of breath, or the need for ICU or ventilation. Shortly after the initial review paper, Mark suggested that we should do a wider comparison between Omicron and the older variants. We had stored samples from the beta and delta waves and already collected micrograph data from our previous COVID-19 publications. So we now collected Omicron positive samples from our clinical collaborator, Dr. Arneaux. Our team of international collaborators wrote a paper where we then compared our South African data between older variants, Omicron, as well as healthy participants. And here you can see some of the outcomes. In this visual here is a visual representation of clot number and distribution. Now, with our knowledge of acute COVID, we also turned our attention to long COVID. Already early in 2021, we found long COVID patients in South Africa. We looked at blood samples from patients and soon realized that there are persistent microclots and widespread endothelial and platelet dysfunction in these patients too. These pathologies may be central in causing the widespread symptoms that these patients may have, and this may lead to tissue hypoxia. We initiated a South African long COVID registry in 2021. And here are some of the symptoms noted by these patients: high blood pressure, high cholesterol, rheumatoid arthritis, previous blood clots, diabetes, cardiovascular disease with some of the comorbidities. Most prevalent of the comorbidities we thought were the fact that cardiovascular disease are central in these comorbidities. Here are some of our results from participants with long COVID from our platelet samples. On the left, you can see platelets from healthy patients, and you can also see we compared it to those of long COVID that are on the right. Large platelet clumps can be seen on the right from patient samples from long COVID patients. We also planned an experiment where we looked at proteomics of healthy plasma, type 2 diabetes, acute COVID, and long COVID. And we added our diabetes samples, as we obviously know that diabetes are more prone to severe COVID-19 symptoms. For the proteomics analysis, we prepared platelet-poor plasma from citrated blood, followed by a trypsin digestion step. To our surprise, we found a visible deposit at the bottom of the tube in the acute COVIDs and long COVIDs, but not in the diabetes samples and also not in the control samples. Plasma proteins, therefore, were fully digested in controls and diabetes, suggesting that trypsin could degrade all or then at least most of the plasma proteins, both in controls and diabetes. However, for proteomics, we needed to further digest the samples, and we then developed a second trypsin digestion step that could indeed digest the sample. We looked at the supernatant after the first trypsin, and we wanted to see if there were still these clots remaining, the microclots remaining. And to our surprise, we found that plasma of patients with type 2 diabetes and plasma from healthy patients, we didn't find any clots, and the samples were clear after the first trypsin digestion step. See the micrographs on the top left. In contrast, microclots in plasma from patients with both acute COVID and those with long COVID still did have microclots after the first trypsin digestion step. And you can see the results here in this micrograph. When we did our proteomics analysis after the second trypsin digestion step, we detected various inflammatory molecules that were substantially increased inside the now digested microclots of patients of both acute COVID and long COVID versus the equivalent volume of the digested fluid of the control samples. Of particular interest was the substantial increase in alpha-2 antiplasmin and the various fibrinogen chains that were trapped in the solubilized fibrinolytic resistant microclots. Our proteomics results also found numerous inflammatory molecules, as I mentioned, and this result suggests that it may affect clot breakdown, and particularly alpha-2 antiplasmin affects plasminogen-plasmin interaction. The lack of breakdown of these clots may have significant clinical value. Here is a simplified diagram to explain clot formation and then breakdown during acute COVID. During acute COVID, spike protein induces microclots that are more resistant to clot breakdown. In some individuals, during the recovery process, healthy fibrinolysis will happen, as it should, and the individuals will return to health. However, in some individuals, about 10 to 30%, healthy fibrinolysis will not happen. And it is these patients that end up having long COVID. A lack of breakdown of fibrinolysis in long COVID patients may result in a persistent, failed fibrinolytic system. We just completed a larger proteomic study where we looked at 100 long COVID patients and 30 controls. This paper is currently under review. We also noted again significant findings of these trapped molecules. And we also found significant presence of antibodies entrapped inside the microclots. Our results again confirm the presence of inflammatory molecules that may contribute to a failed fibrinolytic phenomenon in these individuals. Pathologies noted in post-viral syndromes have a definite golden thread running through them. Our findings suggest that hypercoagulation and vascular damage are key role players causing various symptoms. We wish also to turn your attention to one of the US government documents where microclot presence, viral persistence, and autoantibodies were recognized as central pathologies to look into in finding answers to long COVID. We are also happy to report that our work was featured in June 2022 in Science. And with that, I hand back to Asad.
Well, thank you, Resia, so much for, first of all, the overview of your work and in particular, your latest preprint, shedding light on the differences that you've noticed with Omicron and some really spectacular proteomic results there. So the message I'm getting from your latest preprint is that, as you've already demonstrated, the blood of acute Omicron patients demonstrates anomalous microclotting, although not to the same extent as in beta or delta variants. And also that the difference in the degree of clotting provides a plausible explanation for the relatively low severity of Omicron. May I ask you, whilst in the paper you haven't explicitly talked about platelets in Omicron, I'm curious as to whether you did examine this in some patients, and if you did, what did you see?
Yes, definitely, Asad. We did look at platelets from these patients, and initially we did include it in our paper. However, the co-authors decided we should explicitly focus only on TEG results. But I could perhaps just tell you what we found. As with the previous variants, platelet was significantly hyperactivated in these patients as well. And we believe that it might contribute to vascular damage. So definitely, although the microclots were much less than in the previous variants, platelet hyperactivation were indeed present. We just focused on the TEG specifically, as well as microclots for this publication.
Okay. And I'm guessing that your controls were historic controls pre-vaccination. But did vaccination factor in your disease groups at all? Was that looked at, and do you think it influenced the results in any way?
I think vaccination can influence the results, and we specifically chose our controls so that they were in fact healthy, and a long time after they were actually vaccinated, to make sure that we combat any problems that might arise from vaccination. So it's definitely a question that is an interesting question, and we just currently received South African MRC grant funding to look at differences between long COVID specifically and vaccination. So it would be interesting to look at vaccination as well.
That's really fascinating, because I think that's a question a lot of people are desperate for answers to. I'm also wondering whether you've been looking at BA5 blood, because this seems to have a relatively greater virulence than the previous Omicron lineages, and whether you've noticed any differences there.
We actually have not specifically looked at blood from this more virulent Omicron, but definitely it is one of the next research questions, because I have noted that people have been reporting to be much iller with this variant, especially in other parts of the world.
Yeah. Well, thank you very much, Resia. And yes, I mean, I just thought it would be worth putting it out there that even though Omicron may be mild acutely, because of the sheer number of people affected, it's still a serious problem, both with respect to acute illness and also with long COVID. So our final speaker is Professor Douglas Kell, who is the head of the Bioanalytical Sciences Group at the University of Liverpool. He holds a research chair in systems biology within the Department of Biochemistry at Liverpool University. He is the recipient of a huge number of awards, including DSCs from several universities, and he was also elected a Commander of the Order of the British Empire in New Year honours in, I think it was 2014. He will talk about ischemia reperfusion injury and its link with a symptom found only in ME and long COVID. Certainly one of my most disabling, post-exertional malaise or post-exertional symptom exacerbation. So welcome, Doug.
So just to segue into what I'm going to talk about, there's a saying in science, when a thing was new, people said it was not true. And when its truth could not be denied, people said it was not important. So I think the Omicron results show that it's very likely that not only is it true, but it's important because it must be on the disease pathway if it correlates so well. So I've just put up again some pictures shown in a slightly different way of acute and long COVID studies with the microclots staying with the thioflavin T. And it's very obvious that these are quite prevalent, and they're typically in the range, as it notes there, 2 to 200 microns. So we also recently looked at people with myalgic encephalopathy / chronic fatigue syndrome who bear many of the similar symptoms to people with long COVID, and indeed found the platelets were much more, the microclots were much more prevalent in people with ME/CFS compared with the healthy controls. Again, mostly pre-vax, but we did, click on this slide. We did, as Asad recognized, if vaccination were to induce microclots, which it almost certainly does with people having vaccine-induced injury, that could be a confounder. But we've encoded the vaccination status here, and that seems not to be the case in this relatively small number of people involved in this study. And we did it before adding thrombin and after adding thrombin as well, and broadly similar results. So the consequence of the microclots, the reason that intrinsically they're more resistant to being got rid of, which is why they hang around and are visible, is because of their amyloid nature, before adding in all of the things that Resia talked about, such as the presence of antiplasmin alpha-2 and so forth, that make them even more resistant to the natural kinds of being removed. So being in the range 2 to 200 microns, they can almost choose which microcapillaries they go into and block up, and they'll just find the one that is perfectly the right fit for them and tend to block it up such that the blood cannot flow, erythrocytes can't penetrate well into the microcapillaries, resulting in ischemia, which leads to hypoxia, which immediately and of itself gives a ready explanation for fatigue, which we'll look at briefly, and the likelihood that any tissue that is undergoing hypoxia can have damage. And although it might have been thought odd that you could have an almost unitary mechanism for explaining the many, many different symptoms of many different characters of long COVID in particular, we think that the microclots can actually explain almost all of them and have a role in this. And what I'm going to talk about now is something that Resia and I have been looking at as the consequence of the microclots and the recognition, seemingly for the first time, that what they may actually also be inducing is ischemia-reperfusion injury. So ischemia-reperfusion injury is extremely well known in acute circumstances, such as after a heart attack or a stroke, or indeed during the transfer of organs that are going to be transplanted into someone else. But it doesn't really seem to have been understood as a commonplace in long COVID and indeed ME/CFS. And we think the evidence for this is actually very great. And because it hasn't been discussed before, we wrote a big fat review, which has been accepted in the Biochemical Journal, not quite out yet. And I'm just briefly going to rehearse the kinds of arguments that we put in there. So as soon as you go ischemic, there's a drop in oxygen. Of course, mitochondria can't then make ATP by aerobic respiration, and consequences that they are forced to do glycolysis and produce lactate, which can be measured. But in the reperfusion state, when oxygen comes back, of course, they can start making ATP. But the problem is that they also produce reactive oxygen species. And that's really the big problem of the whole ischemia-reperfusion injury thing. So if you look at the standard mitochondrial respiratory chain, normally complex 4 cytochrome oxidase uses a 4-electron reduction to take oxygen to water. And no troubles are caused as a result. If, however, during the ischemic case, all of the electron transport carriers, cytochrome B and C in complex 3, succinate dehydrogenase complex 2, NADH dehydrogenase in complex 1, are over-reduced. When the oxygen comes back, it can react not only with cytochrome oxidase, but with those other two things. So if oxygen reacts with cytochrome BC1, with a 2-electron reduction, it will make peroxide. And if it reacts with an NADH dehydrogenase in a 1-electron reaction, it'll make superoxide. And these together, which with what we'll see in a second, are reactive oxygen species, and they come as a blast of their production during the reperfusion phase. And if you have unliganded iron present, which you almost always do because cells die, release ferritin, which is an intracellular molecule. It's got nothing to do with iron transport in plasma, although people measure it as such. It's a cell death marker. Free iron in the Fenton reaction at the bivalent state will react with hydrogen peroxide to make the very damaging hydroxyl radical, which will react in nanoseconds with anything that is nearby, causing the Fe2 to go to Fe3. And in the Haber-Weiss reaction, the Fe3 can react with superoxide, which, as we've seen, is also produced to go back to oxygen, but also to return the iron to its divalent state, so that together these reactions act catalytically. So you don't need much to start spewing out large amounts of hydroxyl radicals that are basically going to kill cells. And that, we think, is the cause of a lot of the trouble, especially in so-called post-exertional malaise or post-exertional stress exacerbation, where there's typically a gap of about a day between the exercise which induces the ischemia, followed by the reperfusion, and then the cells take approximately a day to die, and that is the consequence of why you have to wait a day to see these kinds of phenomena. And the kinds of evidence for the fact that there are these ischemic events taking place, which are necessarily going at some point to be followed by reperfusion, otherwise everything will die if there's no oxygen at all going to tissues, are actual measurements of venous oxygen levels, the fact that you do see many more thromboses and aneurysms in patients with these syndromes. We think that COVID toes, where you can actually visibly see, and also sometimes you can see in the retina, anomalies in the flow of blood are fairly clearly also due to that. And as I've mentioned, lactate is produced and can be measured. And although you can't measure the hydroxyl radical, as I said, because it reacts so quickly, it can also react with nitric oxide, and that is a nitrosating reagent. And so nitrotyrosine is a molecule that is classically illustrating the production of hydroxyl radicals and can be measured by reacting with proteins, and equivalently the other macromolecules, hydroxyguanine coming from nucleic acids and malondialdehyde coming from lipids. These are all raised in these kinds of diseases and are kind of markers for ischemia followed by reperfusion. I mentioned the cell death kinetics of that. And of course, part of the argument for the role of the microclots in these diseases is the fact that there are, as yet, non-RCT based studies, but many anecdotal or actual simple performances of patients being given these things, such as clot busters and anticoagulants and antioxidants, which help with the reactive oxygen species and help apheresis where these microclots are removed. So we think in general terms that the evidence for not only the existence of microclots being well established, because you can simply measure them by the methods that Resia described, but we find that it's particularly true for acute and long COVID, and they are of a different character because of their resistance to proteolysis. And they're caused by, in the case of long COVID, a direct interaction of the spike protein with fibrinogen during the clotting process, which is going on all the time. That's been measured in vitro by using recombinant spike protein. So there's really not much doubt about the mechanism. And the fact that they are so resistant to proteolysis is the cause of them hanging around long enough to go and cause trouble by getting stuck in the microcapillaries. So in my part of the talk here, I've rehearsed our view that a chronic version of ischemia reperfusion injury is a significant contributor to the symptoms found in long COVID, and I rehearsed some of the evidence that I think is consistent with this, which also, of course, gives you the opportunity to consider novel treatment options, whether they be for the microclots or antioxidants to deal with reactive oxygen species. And with that, I will thank you for your kind attention.
Well, Doug, that's a perfectly plausible explanation, isn't it, for post-exertional symptom exacerbation? May I ask how the penny dropped as to this potential mechanism?
Because I've known about ischemia reperfusion injury for a long time. And actually, the way Resia and I first met on the internet was through a shared interest in the metabolism of iron. So iron, and in 2010, I wrote a quite long review called Iron Behaving Badly, where unliganted iron appeared in all kinds of chronic inflammatory diseases and indulged itself in the Fenton reaction and the Haber-Weiss reaction. So it was clear that this was quite a widespread phenomenon. We then moved on to recognizing that what it could also do is to allow normally dormant microbes, or bacteria in particular, to grow and wake up and then shed inflammagens such as in gram-negatives like polysaccharide, highly inflammogenic molecule, and in gram-positives lipoteic acid. And so when we discovered the amyloidogenic nature, we actually induced it in vitro by adding minuscule amounts of lipopolysaccharide, because that had come out of the recognition. So the sort of intellectual story moves from iron and the Fenton reaction to iron, the Fenton reaction, and dormant microbes as the cause of all of these troubles. And we could do it with one molecule of lipopolysaccharide per 100,000 fibrinogens. So as noted, the protein, when clotting into this anomalous form, is much more stable, which of course is consistent with it being hard to get rid of too. But from a thermodynamic point of view, there's not an issue. You don't need anything like stoichiometric amounts of the thing to cause these effects.
I mean, it's a perfectly physiologically plausible explanation, although I do have to point out that there have been some patients with ME where clots haven't been found. And one patient in particular springs to mind, and he's been quite public about this actually. So one does wonder if there may be other mechanisms such as, for example, intrinsic mitochondrial problems or parasympathetic failure at play in some patients. Looking forward to the paper coming out.
Not impossible. Don't forget, of course, that the clots may not be found in the venous, the large vessels of the plasma where we extract our blood from to do our measurements. They may be all hidden away and stuck in the microcapillaries, where, of course, we don't sample.
Excellent point. So I guess with that, I have to thank all of our speakers for, first of all, the hours that they put in to come to these results and conclusions. I can't imagine the effort that's gone in. And also to the audience for listening, and hopefully see you again on another one of these.